This article, titled Neuronal Cytoplasmic Inclusion Bodies in the Brain of Lagotto Romagnolo Dogs, a Qualitative and Quantitative Histologic Evaluation, investigates the presence of unusual structures called inclusion bodies within the brain cells of a specific dog breed, the Lagotto Romagnolo. These inclusion bodies are essentially clumps of accumulated cellular components, often proteins, that haven't been properly cleared away by the cell's internal cleaning mechanisms. While they are a hallmark of various human neurodegenerative diseases, their role in dogs, particularly in Lagotto Romagnolos, remains unclear. This study aimed to shed light on the nature of these inclusion bodies and their potential connection to neurological health in Lagotto Romagnolos. The researchers examined brain samples from 23 deceased Lagotto Romagnolos, meticulously documenting their medical histories and conducting microscopic analyses to identify and characterize the inclusion bodies. Their investigation delved into the clinical, histological, and even ultrastructural details, employing techniques like electron microscopy to visualize the intricate structure of these inclusions. The study included Lagotto Romagnolos over 3 months of age with documented clinical and neurological examinations before euthanasia. The researchers examined 7 specific brain areas: the cerebral cortex, caudate nucleus, geniculate nucleus, hippocampus, substantia nigra, cerebellum, and medulla oblongata. Figure 1 illustrates cross-sections of the dog brain at different levels, highlighting these areas with black circles. Researchers further narrowed their focus to 6 Lagotto Romagnolos with neurological signs and 6 without for a quantitative assessment of the inclusion bodies in specific brain regions. The researchers discovered that these inclusion bodies, abundant in the thalamus, geniculate nuclei, and Purkinje cells of the cerebellum, were primarily composed of proteins rich in basic amino acids. Interestingly, the inclusion bodies lacked characteristic markers of typical protein-related diseases seen in humans, such as those found in Alzheimer's or Parkinson's disease. Figure 2 showcases the histochemical and ultrastructural features of the neuronal inclusions. Panel A displays brightly eosinophilic, well-defined, and smooth cytoplasmic inclusions. Panel B shows inclusions staining deeply basophilic with phosphotungstic-acid-hematoxylin. Panel C reveals an absence of periodic acid-Schiff, PAS, positive reaction in the inclusions, whereas cytoplasmic lipofuscin acts as a PAS-positive internal control. Finally, Panel D demonstrates through transmission electron microscopy that the inclusions are not membrane-bound and consist of electron-dense and finely granular material, including small vesicular profiles. The presence of inclusion bodies in the cerebral cortex was particularly noteworthy, as it was significantly associated with the absence of neurological symptoms. This observation led the researchers to propose a fascinating hypothesis. Could these inclusion bodies, rather than being detrimental, actually play a protective role in the brain? This notion challenges the traditional view of inclusion bodies as purely pathological entities and opens up a new perspective on their potential function. Figure 3 uses bar charts to depict the distribution of inclusion bodies across different brain regions in both symptomatic and asymptomatic Lagotto Romagnolos. The figure highlights the number of dogs exhibiting inclusion bodies at specific sites, along with the percentages of symptomatic and asymptomatic dogs with inclusions at each location. Notably, the figure visually reinforces the observation that inclusion bodies in the cerebral cortex are predominantly found in asymptomatic dogs. The researchers acknowledge that several factors beyond the scope of this study could influence the formation and distribution of these inclusion bodies. Factors like gender, inbreeding, environmental influences, and the dog's lifestyle may all play a role, warranting further investigation. In conclusion, this study provides compelling evidence that the presence of these protein-rich inclusion bodies, particularly in the cerebral cortex, might be linked to neurological well-being in Lagotto Romagnolos. While further research is necessary to definitively confirm their protective role and understand the underlying mechanisms involved, this study marks a significant step forward in our understanding of these enigmatic structures and their potential impact on brain health. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.